Anthony van Leeuwenhoek in 1675 saw bacteria for the first time with his crude microscope, which could hardly magnify things up to 150 times. They are distributed worldwide except in the pits of volcanoes, deep strata of rocks, or blood of normal animals. They are present under the icebergs as well as in the hot springs. On the basis of their morphology, the bacteria are divided into the following forms. Number 1, bacillus. These are rod-shaped bacteria. They are further subdivided into many groups. For example, diplobacillus, that is bacillus bacteria in pairs. Palisade bacillus, they are bacillus bacteria stacked one above the another. Streptobacillus, that is bacillus bacteria in long chains. Coccus, these are spherical bacteria. They are further subdivided into subgroups such as monococci, that is cocci occur singly, diplococci, that is cocci occurring in pairs, and tetracocci, that is cocci form groups of 4 cells. Streptococci occur in long chains, and staphylococci, that is cocci occur in an irregular group. Spirillum, these are larger elongated spiral rods. Vibrio, these are half-turn curved rods. Now, on the basis of this staining, they are classified as gram-positive and gram-negative bacteria. Bacteria which retain gram stain during the staining even after decolorization are called gram-positive, whereas those which lose stain after alcohol treatment are known as gram-negative bacteria. On the basis of position of flagella on their body, they are classified as atrichous, that is without flagella, monotrichous, one flagellum at one end of the bacterial cell, amphitrichous, one or more flagella at both the ends, cephalotrichous, two or more flagella at one end only, lophotrichous, tuft of flagella at one or both the ends, and peritrichous, flagella are evenly distributed throughout the surface of the cell. Bacteria are basically unicellular, but sometimes they form loose irregular masses of incipient filaments. The average size of a bacterial cell is about 2 micrometers, though the range lies between 0.2 microns to 40 microns. Bacteria have been divided into 10 orders as per the Burgess Manual of Determinative Bacteriology, quoted from the 7th edition. Number 1, Pseudomonadales. Number 2, Chlamydobacteriales. Number 3, Hyphomicrobiales. 4th, Eubacteriales. 5th, Actinomycetales. 6th is Caryophoneales. 7th, Bigietoales. 8th is Myxobacteriales. 9th, Mycoplasmatales, and 10th, Spirochaetales. Like other microorganisms, bacteria can be classified on the basis of the nutritional requirements for carbon, energy, and hydrogen atoms or electrons. Bacteria can be classified as autotrophs or heterotrophs with respect to the source of carbon as nutritional requirement. Autotrophs use carbon dioxide as their principal source of carbon and light or chemical compounds as a source of energy, while as heterotrophs they use reduced organic compounds as a source of both carbon and energy. Since there are only two sources of energy available to bacteria, so they may be either phototrophs or chemotrophs. Former utilize light as a source of energy, whereas later obtain energy from oxidation of chemical compounds, either organic or inorganic. On the basis of the source of electrons, bacteria may be categorized as lithotrophs or organotrophs. Lithotrophs use reduced inorganic substances as their electron source, whereas organotrophs derive electrons from organic compounds. Therefore, bacteria are of 4 major nutritional groups. Number 1, photolithoautotrophs. Second, photoorganoheterotrophs. Third, chemolithoautotrophs. And fourth, chemoorganoheterotrophs. Now, photolithoautotrophs. These bacteria utilize light as a source of energy and inorganic substances as their electron donor, whereas carbon dioxide is the chief source of carbon. Purple and green sulfur bacteria are the exemplars. Photoorganoheterotrophs. This group of bacteria use light as a source of energy and organic compounds as a source of carbon and electron donor. However, some bacteria use molecular hydrogen as an electron donor. For example, purple non-sulfur bacteria and green non-sulfur bacteria. Chemolithoautotrophs. The bacteria of this category use inorganic compounds as source of energy and electrons, whereas carbon dioxide is the chief source of carbon. Sulfur oxidizing bacteria, hydrogen bacteria, nitrifying bacteria, and iron oxidizing bacteria are the exemplars of this category. Chemoorganoheterotrophs. In this group of bacteria, organic compounds are the source of energy, electron donor, and carbon. It includes all non-photosynthetic bacteria. Besides the above mentioned groups which we discussed, some bacteria rely on inorganic energy source and organic carbon source, and sometimes carbon dioxide. These bacteria are called mixotrophs as they undertake chemolithotrophic and heterotrophic metabolic processes. Bigiota is a common example of mixotrophs. Bacteria generally reproduce by binary fission, as true sexual reproduction is absent in them. However, gene transfer occurs through conjugation, transformation, and transduction.